Thank you for the introduction. Good morning, everybody. My name is Stephen from Newcastle University, and today I'm hoping to give you a very quick snapshot into what our team in Newcastle are doing in the field of leukemia using ultra-long nanopore sequencing. So first of all, why are we doing this? Well, whilst a patient with leukemia will present to the clinic with very similar symptoms, what we do know is leukemia is an extremely heterogeneous disease. Everyone will have a different subtype, which is defined by mostly a very singular molecular mechanism. This can range from copy number variations through to gene fusions to single point mutations. And whilst over the last 30 or even 40 years, we've done a fantastic job with standard of care techniques such as G-banding and karyotyping, and we can now determine the subtype in a lot of cases, there are also a lot of cases that are remaining cryptic and we're not able to detect with standard of care techniques. So this is where ultra-long sequencing really comes in. So we set out with the aim to demonstrate the potential of ultra-long sequencing to not only detect, but also fully molecularly characterize these subtypes. So what did we do? So first of all, we've ultra-long sequenced around 32 leukemia research samples. I'd like to point out some of these have sat in a freezer for over 30 years at this point, and we've been able to do this. So we extracted high molecular weight DNA, we put them through a PromethION sequencer, one flow cell per sample, and then we put them through a custom analysis pipeline. Although this pipeline, I say is custom, it uses all publicly available tools. So what are some of our results? Well, very excited to say our average or median N50 is around 82 kb, 80,000 bases. Very, very, very nice. We achieve median coverage of around 24-25x. Our longest read in this cohort is 1.46 MB. Although, just a quick shout out, we have got some in excess of 2 or 3 MB in other cohorts. And we have a 97% sequencing success. So actually, of these 32 samples sequenced, only one of them failed. It didn't fail sequencing, but it failed to achieve the coverage that we required for this cohort. So my favorite example is the DUX4 rearranged cases, and this was mentioned yesterday, but what we're able to do now is not only detect DUX4 rearranged cases, and these are in extremely complex regions. So DUX4 is an extremely repetitive region of the genome. IGH, which you see on the screenshot behind me, is also extremely repetitive. But what we can now do from a multitude of reads is detect very precise breakpoints to base pair resolution for the very first time. So on this example, I think there's 15 reads supporting this. But if we look at one individual read, so this is now looking at just one singular read, which is spanning over 60 kb, we can actually determine the entire structure of this complex variant for the first time. So we have a read here, which is spanning through the IGHM locus. We have an insertion of DUX4 that is around 4 kb in this case. We then detect multiple deletions, large-scale inversions, everything all the way through this locus for the first time. Now, this is one example, but obviously we have multiple examples just among this cohort. But I think it really demonstrates that something we've been trying to do for 30 years and unable to do with other technologies, we can now do almost trivially. We can now look at these extremely complex repetitive regions and characterize these patients for the very first time. So with that snapshot, if anyone would like to come and speak to me, I'd love to answer any questions throughout the networking sessions. And if anyone has any questions, I'd love to take them now. Thank you.